Hello guys, it's me, Angelica, and our lesson for today is all about differential gene action. But first, let's have our learning objectives. At the end of this topic, you will be able to describe the basis of cell differentiation, learn about the process amplification of gene interest. Third, differentiate between the transcriptional and translational control. And third, explain the epigenetics control mechanisms. So let's get right into our first key concept, the different factors that influence how genes are expressed. So this is one of the topics that we are going to explore for today. But first, we have to understand what is cell differentiation. So this is an important topic or important concept for us to understand gene expression. Now, if each of your cells contains the exact same set of DNA, then why aren't cells the same? And why don't they all look the same and have the same functions? Well, that's because what we call cell differentiation. So as we look at this chart here, we have in the middle something called stem cells. And really, their stem cells are going to differentiate into all different cell types. So this is called the cell differentiation. So when we say cell differentiation, it is a process of cells that becoming specialized to a certain cells to differentiate into muscle cells, cancer cells, blood cells, liver cells, and with different functions and shapes. So stem cells, like I've said, are cells that are not yet specialized. So they can still become anything that they want to be. So they have the ability to become all these different types, which is why you hear a lot from stem cells research. And why that's such a promising field if we can get these stem cells to divide and differentiate into anything that we want to be. And it can be also help when treating people with certain diseases. But how do stem cells decide what to become? Well, that's when we come up into idea of gene expression. So genes are the light bulb and the switch is the express control. Now when the light switch is in the on position, it means that genes are expressed. On the other hand, when the light switch is in the off position, it means that genes are not expressed. The DNA is what holds the code that determines which genes will be expressed to make a cell specialized. So in the code of DNA, some says that genes are going to express into light switch on, and this going to be expressed light switch off or not expressed. So the RNA carries the code from the DNA to the ribosomes where the proteins are made. Now if certain genes are expressed, then certain proteins will be made. However, if certain genes are not expressed because of the light switch off, then proteins will not be made. Other things can influence gene expression. Some environmental factors can influence which genes get turned off and which genes get turned off. Or they can mutate or alter DNA. And these factors either be the internal factors and the external factors. So looking at the couple pictures right there were a hinting at graduation of being an external, and on the other hand, internal is being something like a diet. So external factors is something like an alcohol. So we look this a fetal alcohol syndrome. Now when the fetus is exposed to alcohol while in the womb of the mother, now this is not even a small amount of alcohol. This is a certain amount of alcohol that the fetus exposed to a certain symptoms that can include physical and mental abnormalities. So you see a picture there of a child who had fetal alcohol syndrome because she was exposed to much alcohol while in the womb of her mother. So you see all these different things like small head, low nasal bridge, and these are the factors that which are affected by the genes was expressed widely when the fetus was in the womb of her mother. So let's look at the internal factors. So hormones are certain proteins that produce body that can change the way that the genes get turned on and off. So the great example of hormones that affecting the gene expression is the puberty. When you go through puberty, hormones are released, certain amounts and some more than the others. And they influence which genes express at what time and how the process of maturation goes about. So let's move on to our second subtopic, which is all about the gene amplification. So let's talk about the recombinant DNA containing the gene of interest is amplified. Well, the gene of interest is amplified by using the process of PCR, or what we call polymerase chain reaction. So when we say PCR or polymerase chain reaction, it is a special technique in which multiple copies of gene interest are synthesized by draw. So there are some requirements of PCR or polymerase chain reaction. These are the DNA template, nucleotide primer, and the enzymes. So the first one is the DNA template. So it is the largest DNA that needs to be amplified. And the second is the nucleotide primers. Primers are complementary to the strands of target DNA or the DNA template taken. Primers for the process of PCR, it is the two sets of nucleotide primers that are required. And the third one is the enzyme. For the process of PCR, the enzyme of DNA polymerase is taken. Now that we know the basic requirements of PCR reaction, let's also learn about the working of PCR. There are three main steps of PCR or polymerase chain reaction. These are the denaturation, annealing, and extension. The first one is the denaturation, when a double-stranded DNA template is separated into two single strands. The second one is the annealing, when the temperature is lowered to enable the DNA primers to attach the DNA template. The last step would be the extension, when the temperature is raised and the new strand of DNA is made with the polymerase enzyme. So this is how the target DNA gets amplified. When all the three steps are completed, the first cycle of PCR are also completed. When these cycles are repeated, many times a DNA segment is amplified by 1,000,000 times. So the third subtopic that we are going to discuss is all about the transcriptional and the translational control. So let's get right into our first key concept, which is the protein synthesis. Protein synthesis is a process of making proteins using instructions of DNA or those sections of DNA what we call genes. The central dogma to all of biology is this picture right here. DNA is transcribed into messenger RNA, which then translated into protein. Proteins control the number of the things that we do in our bodies, including the enzymes and the hormones. Then why are proteins important? The first one is that they catalyze biochemical reactions in the form of enzymes. Second, they help to fight diseases in the form of antibodies and also transmit messages in the form of hormones. And lastly, they also build bones and muscles. So the protein synthesis major processes are the transcription and the translation. When we say transcription, it is when you go from DNA to messenger RNA. On the other hand, translation is when you go from messenger RNA to protein. So let's know about the difference between the DNA and RNA. The sugar is what's slightly different from DNA to RNA. We have deoxyribose sugar for DNA and ribose for RNA. Also, in nitrogen bases are slightly different. So in DNA, we have the ATCG, while in RNA has no thymine. So there's no T. However, it's replaced by letter U, which is called uracil. The DNA is double helix, while the RNA is the single helix. DNA is located in the nucleus. On the other hand, RNA is also located in the nucleus and cytoplasm. So here's the overview of what they look like. There are types of RNA. The first one is the messenger RNA that carries copies of DNA. The second one is the transfer RNA, in which it transfers amino acids. And lastly, the ribosomal, in which it makes up the ribosomes. This is how they look like. These are the messenger RNA, transfer RNA, and ribosomal RNA. Transcription is a section of DNA or genes, and once it is copied to a messenger RNA. So there are three steps of transcription. The first one is that DNA is unwound of a gene. The second one is the RNA polymerase copies the DNA strand, creating complementary to messenger RNA strand. And the last step is the messenger RNA moves from nucleus to cytoplasm and binds with the ribosomes to begin translation. Let's move on to the translational control. When we say translation, it is the information from messenger RNA is used to make a protein. Also steps in translation. The first one is that messenger RNA attaches to the ribosomes at the start codon. So when we say codon, it is a sequence or triplet sequence of these new nucleotide bases. The second one is that the transfer RNA carries the amino acid specific on the codon on messenger RNA. And lastly, the amino acids are bonded together to make a polypeptide. So let's move on to our fourth subtopic, which is all about the epigenetics control mechanisms. Our genetic code or DNA is directed, controlled, and regulated by the epigenetic factors which surround scaffold DNA in order to work straight the cellular mechanisms of life. The epigenome is the collection of DNA, RNA, proteins, and their chemical modifications that alter gene expressions without changing genetic code. Discovery research in epigenetics seeks to understand how the environment influences regulation of genes packed within our chromatin, resulting into diseases such as cancer. Epigenetic machinery modifies accessibility regions in DNA by adding marks to the tails of histones to create epigenetic patterns of chromatin modification. The addition of methyl group to a histone tail changes the electrochemical charge of the histone, causing it to relax and release the DNA. This makes the DNA in that region more accessible through transcription machinery and allows the gene to be expressed. Placing a methyl group of histone tails can either increase or decrease that pattern of gene expression depending on where the mark is placed. Additionally, placing a methyl group directly on the DNA results in the permanent shutdown in that genetic region. The epigenetics pattern modification of chromatin change during development and the differentiation in response to environmental changes. When cells are exposed to environmental stimuli, the patterns of chromatin modifications become unregulated as cancer cells hijack the genetic molecular machinery. More half-on-human cancers contain mutations in DNA-associated proteins that involve in chromatin organization and regulation. So I think that's all for today, and I hope you've learned about so many things about my discussion. Thank you and God bless.